Hello and welcome to my channel all about biotechnology. Today I'm going to discuss with you a very important aspect in molecular genetics, that is genetic complementation. We will very systematically learn what is this genetic complementation is and how it has helped in developing wealth of knowledge about the various metabolic processes, how many genes are involved, and how genes are regulated. Although I will focus on understanding the basic aspect of genetic complementation today. So before I proceed, a request that if you have not subscribed my channel, please do so and like the video. Okay, so what is genetic complementation? Before we start dissecting and understanding genetic complementation, let us understand what is complement, which is a noun from where complementation has been derived. So in English, complement means something that is complete or makes it perfect. So that is what we understand by word complement. We are looking at something which is perfect or makes it perfect. Okay, coming to complementation or genetic complementation. Now, we have to understand that this event or process takes place across the biological spectrum of living beings, from eukaryotes to prokaryotes. But of course, its discovery was made in prokaryotes. And when we talk about living world, we have phenotypes. Each living organism has certain characteristics on the basis of which it is distinguished from other organisms and within the organisms. And that is what we call phenotype of an organism. Okay. And this phenotype is determined by a set of genes. Okay. So that is what we call wild type phenotype. A perfect, the bunch of perfect features of an organism, which is taken as wild type. Okay. Now, there are variations to this wild type phenotype. That is when a phenotype is not expressing or wrongly expressing. How that happens? That is because of the mutations. So if a phenotype has 3 genes, for example, and one of the gene gets mutated, then what will be the impact of this mutation? That the phenotype will not be expressing itself. You won't see that phenotype, you know. Now, when there is another mutation, a second mutation, and the second mutation is such, which is correcting the first mutation, that will lead to the organism regaining the wild type phenotype. Okay, so complementation is the result of 2 mutations which are complementing each other to make the organism perfect. That is to make the organism wild type. Okay, so to define what is genetic complementation, basically it is the complementation of 2 mutations coming together for resumption of a wild type phenotype. Okay, so it means, if we take an example of a prokaryote, and that's where I would be focusing on, And that's where this phenomenon was first observed. Okay, so let us understand this aspect which I had been talking about with an example. Okay, so if a bacterial cell is able to synthesize proline, which is an essential amino acid, the phenotype is pro plus phenotype. Okay, and you may be knowing, or else I'm revising with you, the proline in E. coli is synthesized from this substrate, what we call alpha-ketogluteric acid. This is the starting point. And then it involves three genes, Pro-A, Pro-B, and Pro-C. So this phenotype is because of the presence of these genes, and these genes are perfect and they are expressing themselves. They are making these enzymes, like Pro-A is responsible for this enzyme we call glutamyl kinase, which converts glutamate to glutamyl phosphate. And then Pro-B codes for GSA dehydrogenase, which converts gamma glutamyl phosphate to gamma glutamate semialdehyde. And then it undergoes a chemical reaction, cyclizes itself to make it to P5C, pyrroline 5 carboxylate. And then this is the third gene, which is Pro-C and codes for an enzyme, pyrroline 5 carboxylate reductase, which reduces this P5C to proline. So that's the total pathway. And in fact, this pathway was deciphered making use of this genetic complementation, a process. Okay, and an assay was developed, what we call complementation assay. Okay, come back to this. So when a cell is pro plus, it means all the genes, pro A, pro B, or pro C, are perfect. They are functioning. They are making a functional protein, which is doing their job and making the bacterial cell a pro plus cell. Now, imagine if there is a mutation in one gene or two gene or all the three genes. What will be the impact of this mutation? If the mutation is in Pro A, this gene means this product will not be made and this pathway will be stopped here itself. And the result would be a pro minus phenotype. So you have a wild type where all sets of genes are functioning all right, and it gives you a phenotype. So if there is a mutation, like if there is a mutation in Pro B gene, again the pathway is going to stop here. It won't proceed further. And the result would be a pro minus phenotype. Similarly, if there is a mutation here, then again the result consequence would be that the process will stop here itself. It won't be reduced to proline. Okay. So the point I'm trying to make here is that you have a wild type phenotype, and because of a mutation, either of in any of these genes, in one gene or more than one gene, the consequence is that you have a mutant phenotype, which is a pro minus phenotype. Okay. Now, this complementation process is seen in a diploid cell or a heterokaryon. It means you have to have sets of two alleles of the same gene or two copies of the same gene in the same cell. Okay. For example, if in a cell you have the original genotype where A is mutant or A got mutation and it's not functioning, and you have a pro minus phenotype. Now, if in the same cell you have, or in bacterial cells, as you may recall, we have partial diploids or merodiploids. So in a merodiploid situation or a partial diploid situation, if we have a gene which is perfect, which is right, So what will be the consequence of the presence of this perfect gene in the same cell? That this gene, although it is not functioning here in the chromosomal DNA, but in the extra copy, the intact gene is there, and that is going to make this protein, and it will help in regaining the wild type phenotype. And that is what precisely happens in complementation. That two mutations, they complement each other. Means one mutation is making it non-functional, and with the help of another copy of the gene which is perfect or which is right, it is complementing the defective gene, and you regain, or the cell regain, the wild type phenotype. Okay, so that is the crux of the genetic complementation, that uh, you have a mutation in a wild type cell, which renders it to losing that phenotype. And because of the presence of an intact copy you see of the same gene in the cell, in a partial diploid situation, in a mirror diploid situation, you regain the wild type phenotype. Okay, and that it means, what is the conclusion one can draw? That if the two mutations are complementing each other, they are not in the same gene. Means in the partial diploid, this gene may be intact, but there the mutation may be in the other gene in the partial diploid. But that mutation is complemented by the intact copy in the chromosomal DNA, and the mutation in the chromosomal DNA is complemented by the intact copy which is present in the extra copy. So both are mutated, but though the two mutations are complementing each other, it means they are not in the same gene. So that is one conclusion. If the two mutations are not are complementing each other, it means they are not. It means they are in the different genes. Okay. So this is the basis of understanding genetic complementation. Okay. Now let us look at another example. Okay, very carefully. Suppose you have a mutation, a mutant that is pro minus. Okay, that is pro minus. Okay, but we don't know where the mutation is out of the three gene A, B, C. So different set of partial diploids or mirror diploids were generated. Okay, for example, one set of partial diploid is this. This is the chromosomal genotype. Okay, and this is the partial diploid genotype, wherein you have B minus, A plus, and C plus. Okay, and in the mutant, where we don't know where the mutation is, so if you look at this cell where you have and the result is that it regains the wild type phenotype. That is, see, it is B minus, but it is B plus here. So it is regaining the the wild type phenotype. Okay, so it means this is B minus, but this is B plus and this is B minus. So the function of B gene is contributed by the gene which is located on the chromosome, and remaining A and C are intact in the partial diploid. So there is complementation happening here. The B is complemented by the gene which is located on the chromosome. Similarly, C, if you come here, this mutant is C plus actually. And partial diploid which was generated, and we knew where the mutation is, B plus, A plus, and C minus. And the phenotype of this is pro plus. So it means all the three genes are functional. Okay. Although in the partial diploid set of genes, we know it is C minus, but since we are getting pro plus phenotype, it means uh, a defect in the C is complemented by the chromosomal gene, which is intact. Okay, so it is able to regain the wild type phenotype. Okay, so again here the function of C gene is complemented by the gene which is located on the chromosome. Now, coming to this, where it is A minus, okay? And what happens in this cell where the partial diploid is generated, where we know it is A minus. And this cell does not regain the wild type phenotype. This does not regain the wild type phenotype. So it means the mutation is in A. But I mean, in the chromosomal DNA, this gene is mutated, and also on partial diploid it is, and therefore there is no complementation, and therefore the cell remains pro minus. Okay, so in the conclusion, what we are seeing here, that in two situations, the cell regains the wild type phenotype here and here, but in third scenario where it does not regain wild type phenotype, it means both the mutation, the mutation in both the set of copies are affecting the same gene, and therefore they are not complementing each other, but the complementation is seen here and here. Okay, so that is the actually, that is the basis of what we call complementation test or complementation assay. And this essay has been employed in last in 60s to generate a whole lot of information about the genes, how many genes are involved in a pathway, and then subsequently, those genes were biochemically characterized, and all the pathways in bacterial systems were, whether it is amino acid biosynthesis or even utilization of carbon sources, lactose, galactose, all the pathways were, the synthetic pathways or the degradative pathways were deciphered by doing this complementation assay or complementation test. So what it creates actually is the complementation blocks. So you can identify that for a pathway, how many complementation blocks have been generated. And each complementation block is responsible for one set of product, or you can say the complementation block is a gene actually. Okay, so if you have understood, that is the basic concept behind the complementation, and what all it has contributed is to, as I mentioned, determine the number of genes responsible for a particular phenotype. Okay, and also very important, distinguishing regulatory sites and the regulatory genes. And that's how the concept of lactoperon was generated. And we know how prokaryotic genes are regulated. And following similar lines, you have the eukaryotic gene expression. The basic concept remains the same. So I believe I'm able to make you understand this complex phenomenon in a simple way, and I will stop here and continue with some more basic aspects of molecular genetics in my future videos. So thank you again for patient hearing. And once again, if you have not subscribed, please do subscribe my channel and get benefited by whatever I have understood in biology or in biotechnology.